Hello, everyone. Happy to be here. Thank you for organizing this meeting and inviting me to speak here. So basically, I'm going to, switch gears a little bit and speak about immunology. I guess this is not immunology background here in the audience, so I try to not to get into, very immunological details, but I try at least to convince you that aging of the immune system is playing a major role in the aging process. This is what we are currently understanding. So basically, we have in the lab, what we do is a lab of neuroimmunology. Basically, we study the connection, interactions between the immune system and the brain in various systems. We look at the microglial cells, as Valerie described before, in Alzheimer's and other diseases. We look at the chronic stress and how it impacts, immune regulation, high-fat diets, which impact on hypothalamic aspects and blood-brain barrier. All are very important and key aspects of aging. So what I'm going to talk today is primarily about changes in the immune system with age and how they impact the aging process. So first of all, we all know that the population structure is changing, but most important is to look at, age-related diseases, as we have heard before. And I think that one important note to take from here is that age-related diseases do not begin, at the 70s or the 80s. They begin at the fourth decade of life. And I hope that I can convince you why it happens and why we can look at the immune system as a way to basically diagnose and predict this pace of aging. So as you can see here, in fact, cancer, obesity, and diabetes, they begin at the fourth decade of life, and these are already considered age-related diseases. So when you speak about aging, I think we need to define, when aging actually begins. So one way actually to look at the aging process, and I like this slide a lot, because it looks at, very simplistic way of defining aging, which is basically the balance between damage and repair. Okay, so the more damage we have, the less repair we have, the faster is the aging process, and this, of course, can be impacted by genetic factors and environmental factors. So here you see there is unrepaired, damage accumulation, which lead, to senescence, as we have heard in details from Valerie today, for apoptosis. This all can result in inflammation and loss of regenerative capacity of the tissues. On the other hand, we have accumulating somatic, oncogenic mutations, which can lead to the increased risk of cancer. So I think the key question that we ask ourselves now in aging are the following. First of all, when aging begins. This is very essential to understand if we want to begin, treating age-related diseases and improve the health span, as was discussed before, not only the lifespan. And then if we want to know that, we need to know what drives aging, right? Why is it that at some point in life we accumulate more damage that we have done before? And then can we predict the biological aging? Basically we know the chronological age, but we don't know always what is the biological age. And, when you reach to the 50s and 60s and 70s, I think you can find a gap of even 20 years between individuals in terms of the biological age. So this is a key component of the aging that we don't really diagnose these days, and I believe that we can do that by just looking at the immune system. And then can we predict, age-related disease susceptibility? I think this is one of the most difficult challenges, because now I think we have good tools to look at biological aging, but we don't know what kind of diseases we are facing. Each individual in its own story. And so the question is can all these questions be addressed or explored via mechanism of immune aging? And this is what I'd like to show you today. So first of all, we can look at the immune system. The immune system is composed of many cells that come out of the bone marrow, and they are basically circulating in our body and taking care of our body day to day, not only protecting us from infection, they are really important for keeping the homeostasis of the body. Now, a key population in immune systems, which I like the most, are the T cells, which are, came really late in evolution, but they became to be the keystone of the immune ecosystem. Namely, that if the T cells, lose their power, they basically take the entire boat down together. And primarily, we talk about the CD4 T helper cells, as opposed to the CD8 cytotoxic T cells, which basically take care about, viral infected cells, or they are, responsible to kill cancer cells. And all these functions, also killing senescent cells, are very important in aging. But at the same time, we have the CD4 T helper cells, but these cells really orchestrate the entire function of the immune system, including the activation of CD8 T cells and other aspects of the immune system. So CD4 T cells, as I said, are main regulator of all immune functions that we are aware of in the body. And basically, this is a nice review from my collaborator in Spain, Maria, which have shown nicely that, also looking, citing our papers, showing that, two major components of immune aging in lymphocytes. One of them is thymic involution. Basically, the thymus is involuting and give rise to decline in the lymphocyte repertoire. And at the same time, we have mitochondrial dysfunction, and both of them actually lead via various processes that have been defined over the years to cause immune deficiency and inflammation. And these two aspects are sufficient to cause all age-related diseases and mortality, that we face in aging. So, and there are studies, also seminal studies, showing that just manipulating the lymphocytes in mice, you can impact the longevity of the animals. So what I'd like to show you today is, first of all, how the CD4 T cell landscape is changed over the age. So we have done this study a few years ago. We are surprised, that this study was actually the first study showing the landscape of lymphocyte aging in mice. And it was really a breakthrough in the field, because until that time point, we couldn't know what is the dynamic processes of lymphocyte aging over the age. And then we found a unique and puzzling T cell subset that we have not been aware of before, that accumulate with age and impacts frailty and longevity. So the first study actually was in collaboration with Professor Esti Eger-Lochten at BGU, and two very, very talented PhD MD students now doing the residency in hospitals. And what we have done here basically is to take lymphocytes, or primarily CD4, the T helper lymphocytes that I've shown you before. We isolated these cells from young mice at 2 months of age and from old mice at about 24 months of age. And what we've done is single cell RNA sequencing, and basically we could map the differences that we have in the CD4 T cell repertoire. And so what you see here, that in young animals we had primarily a subset that we call naive T cells. These cells have not been experienced with antigen before, so the majority of cells in young mice are naive because they have a very high turnover in the cells, they mature in the thymus, and they basically circulate in our body when we are young, and so the repertoire is really very powerful. So with age, we found the accumulation of various subsets that we call them age-related subsets. Among them are regulatory T cells, which basically suppress immune responses, exhausted cells. These cells basically express molecules that suppress the immune functioning, such as PD-L1 that was explained before by Valerie, but additional molecules. And then a very surprising subset that is cytotoxic CD4 T helper cells. So I've shown you before that the T helper cells basically are not cytotoxic. We have a unique cytotoxic arm within the lymphocytes, and all of a sudden, these mice express or differentiate cytotoxic CD4 T helper cells in aging, and that was very surprising for us. So overall, what we have seen is that CD4 T cells, while they are very homogeneous in young animals, they switch into very heterogeneous population with age. So, and although there are so many variants of cells in aging, you could still see certain trajectories that one could identify. And we basically collected the markers, and we basically found how this process is gradually progressing with age. So it's not a one-step process. It occurs throughout life and basically give rise to various type of dysfunctional lymphocytes. And this dysfunctional lymphocyte, of course, can drive the entire process of aging, as you can identify. So basically what I'll show you here is a summary of this study, because it really brought me to think what is basically going on here, because we know that, and if we look here on two issues of repair that goes down with aging, and basically the damage that goes up with aging. And we can see some variation, of course, between individuals based on genetic background, environment, and so on. So what happens here, and we look here basically at a very young age, at the time that we are actually at puberty, okay, when we have the sex hormones coming up, the thymus is involuting. So this basically suggests that the thymus involuting massively during puberty begins to change the landscape or the repertoire of our lymphocytes. Okay, and this basically follows by the accumulation of dysfunctional lymphocytes. Okay, dysfunctional lymphocytes which lead to chronic inflammation. They also give rise to accumulation of senescent cells and also to reduced stemness of the tissues. So basically what we describe here is that aging begins at puberty because it impacts the repair system of the body. Okay, so once we begin to change the repair system at puberty, then we begin accumulating the cells, and if you follow this timeline, you would see why basically we begin having or exposing to age-related diseases already at the fourth decade of life. So this is the summary, but I will skip that for now because to make sure that I can go throughout the topics that I wanted to cover. And so coming back to this slide where I've shown you that we have this heterogeneity, and we have also the accumulation of cytotoxic CD4 T cells. So this was very surprising to us, as I said before. And you can see that if we look at the transcriptional program of these cells, I mean, they had a very similar transcriptional factors that are expressed by CD8 cytotoxic T cells. And so we asked ourselves why basically we have this phenomenon, why we need cytotoxic CD4 T cells. I can tell you honestly, at the beginning, when I saw this result, I didn't know what they meant, and I was thinking maybe it's Ben-Gurion, properties. Some mice in Ben-Gurion University accumulate these mice for some reason. I really didn't know because this is not in textbook. This is nothing you consider in immunology. These cytotoxic CD4 T cells do not basically exist. So then, lucky for us, 2 months later, there is a study coming by a Japanese group, and they look at supercentenarians. So supercentenarians, as you know, they live long after, even after 100 years, and they have actually even a healthy life. And they did the same study as we have done, basically mapping the lymphocytes, or the CD4, or the T helper lymphocytes, and they found that about 50% of the T helper cells were CD4 cytotoxic T cells, exactly the same ones that we observed. So this is what they basically wrote in the study, that the study reveals that supercentenarians have unique characteristics in their circulating lymphocytes, which may represent an essential adaptation to achieve exceptional longevity by sustaining immune responses to infection and disease. So that was about that, and we said, okay, we have something very interesting here, but what it is, what these cells are doing in aging, no one really knew. And so what we have done is, since you see here the transcriptional machinery of these cells, and e-RMS is one of the transcription factors that is essential for the development of these cells. So what we have done is basically in another study, also guided by Dr. Chezi Eliyahu, is to knock down these cells, so basically prevent the accumulation of these cells during aging. So this is a cross between two different mice, and it allows us to conditionally deplete e-RMS from cells, and hopefully also avoids accumulation of cytotoxic CD4 T cells. And this is what we have done. And basically, we have done a study where we took 20-month-old mice, knockout or control, and we gave them tamoxifen basically to deplete the cytotoxic population of T helper cells, and then we followed the mice. So first of all, lucky for us, we could deplete this population. So this population was no longer accumulating in these animals. And if we look at them at the grip strength test, we could see that while normal mice performed like normal, and these knockout actually were deteriorated. And also if we look at activity and food, so you see that the mice, the knockout, were less active and even eating more. So these are all phenomena of accelerated aging. And also surprisingly, we have seen that senescent cells, all the senescent cells that Valerie was describing before, actually were accumulating in these knockout animals. So depleting the CD4 cytotoxic T cells resulted in accumulation of senescent cells. And basically, we have done also lifespan experiment, and we depleted, again, using the same system, the CD4 T cells, the cytotoxic CD4 T cells, and as you can see, the mice with depleted population of cytotoxic cells actually live less than the littermate controls. So this means that this subset of population is responsible for longevity in this cohort of mice, and to know if indeed senescent cells are the cells that responsible for this effect, we did the same experiment, but now treating the animals also with senolytic drug. And by doing this, we could prevent this effect. And this was actually work that we have done in collaboration with the lab of Valerie. So basically, this is the summary of this study, which I skip, and I would like to address the last question that we ask ourselves: why we need cytotoxic CD4 T cells, why we need T helper cells to differentiate into cytotoxic cells, and why this is so important, keeping in mind that we have the CD8 cytotoxic T cells and we have also natural killer cells in the innate immune system. So basically, again, I'm coming back to the senescence process, and I don't need to detail because Valerie has done it before. And so basically, it's a physiological process, and if these cells are clear, everything is well regulated. But the question and the problem begins when they accumulate, and then you need mechanisms that can clear the cells. So basically, what we have shown is that if lymphocytes differentiate to become these e-RMS cytotoxic CD4 T cells, they can help the clearance of these senescence from the tissues. And the reason is because this e-RMS factor is not only responsible to enhance the cytotoxic machinery, but it also gives the cells metabolic fitness. And this is extremely important for lymphocytes living in tissues with chronic inflammation. They have to survive. And what happens many times for the CD8 cytotoxic T cells is they undergo exhaustion or even they die, and therefore they become ineffective. These CD4 T cells then take, the role and become the effector and important cytotoxic subset that basically can do the job. And these cells can basically kill cancer cells, they can kill senescent cells, they can kill chronically infected cells, and by doing that, they can give rise to extended longevity. So, and this is just to let you know that we do the same thing in human, and I'm just finishing. Basically, this is our working strategy, and I just want to point out the healthy aging phenomenon, which seems to be having a characteristic of adjusted phenotype. Because many times we talk about rejuvenation. Here we are not talking about rejuvenation. These older individuals do not need the immune system of a 20-year-old guy. They need to have an adjusted immune response characterized here by this cytotoxic CD4 subset. And this is, I think, very interesting. And I also would like to note that paradoxically, for the most part of aging, the changes in lymphocytes may affect primarily tissue homeostasis and repair, and less the ability to fight pathogen. And this is basically a mind shift in the way we look at it. So I'll finish here. There are some take-home messages, but we don't have time. Just thanking our collaborators and the team of students in the lab doing the work, and happy to take any questions if there's still enough time. Thank you so much.
Thank you for your talk. How crazy is it to think about vaccine against aging, educating the immune system to attack senescent cells as they appear?
It's not crazy at all. This is, I think, what we should be doing. I think it's crucial. So you can't have long life or healthy long life without a good immune system. So, and when we have to think, and this is what's unique about this study, that we can define what is the properties or characteristics you need for an immune system at different stages of life. So first of all, we have a roadmap where we can define the landscape of lymphocyte at each stage of aging, define how normal or abnormal it is, and how it relates to the biological aging, and then learn how to modify it, to basically acquire the properties that we need. And this is exactly the thing that we are doing and what we need to do.
Thank you. So, do you find any correlation in puberty between hormonal level and this differentiation in the thymus that you have described? And if so, how this correlates with the centurions, which obviously don't have the same hormonal level anymore?
Right. Yeah, that's a thing, very interesting question. We didn't look at correlation. It is very well known that, the rise in these sex hormones give rise to thymus involution. What is very interesting, I think, that to consider a thymus rejuvenation strategy. And actually, we have done it in the same AD mice, Alzheimer's disease mice that Valerie has shown before, and we did find a very dramatic improvement in the function of these mice. So something to note is that at menopause, sex hormones actually go down, and it may actually give rise to some rejuvenation properties of the thymus, and it may has to do with a more, better functioning immune system of women at these ages. So this is something very interesting. Exactly, because as the increase in sex hormones, this is what involutes the thymus. So the reduction in sex hormones give rise to its regeneration. So it's very interesting. It's not investigated, but I think it's very promising. And we have very nice tools actually to rejuvenate the thymus. Yes, there are several ways of doing it, and it may be very promising also as another strategy.
I do like it. In humans, we had the DREAM study, which is a very famous study in longevity medicine. And that was targeting the thymus, trying to rejuvenate it. Is that in line with what you are doing? Is that a very promising longevity therapy?
Yeah, I think so. I mean, I think it's a very, but we still have to, to learn a lot, because what I've shown you today is a very surprising phenomenon that we haven't expected. So to see these cytotoxic CD4 T cells that come at a time stage where the thymus cells was not functioning at all. And yet you have this differentiation of cells which occurs in the tissue. These are terminally differentiated cells. They are not programmed in the thymus and they are not programmed in the lymph node. They are terminally differentiated in the tissues. And the studies that I haven't shown you today, but for the next time, is that if we look at patients with cognitive decline, they basically have deficit in this population. And this may actually be part of the, at least part of the reason why they have cognitive decline. So yes, there is a lot to learn. What is the, the immune systems that we need at different age and for longevity to be able to manipulate it in the ways that we need. But that's, I guess, and I hope it's a direction to move forward.
Thank you very much.